Hello, so this is now the second lecture, my second lecture in the course, and it will deal with membrane protein bioinformatics, that is to say the application of bioinformatics methods in the field of membrane protein structure prediction. So let me share my screen again. So, membrane protein bioinformatics. I will deal with two different aspects here. The first one will be not so much on membrane proteins per se, but rather how membrane proteins and other proteins find their correct location inside the cell. And then I will go on to talk about membrane proteins in particular. So the first issue is to do with protein sorting signals. And what I mean by that is that the cell, especially eukaryotic cell, but also bacterial cell, has a complex interior with many different compartments to which proteins can be targeted, specifically targeted. Now, most proteins, almost all proteins, are initially synthesized in the cytosol by cytosolic ribosomes, and then need to be targeted to the specific compartment where they're supposed to do their job. So obviously, some proteins remain in the cytosol. Some proteins are imported into mitochondria, for instance. Others are imported into peroxisomes, or in plant cells into chloroplasts. And finally, quite a good number of proteins are initially imported into a membrane-bound organelle called the endoplasmic reticulum. And eventually, after transiting through the endoplasmic reticulum and to an intermediate compartment called the Golgi complex, these proteins are largely secreted outside the cell. So they become secretory proteins like hormones and other proteins in the blood, antibodies and things like that. So obviously, proteins have to contain some kind of information that directs their trafficking to the specific organelle where they're supposed to end up. It turns out that this information is, in most cases, encoded in short stretches of the amino acid chain called targeting sequences or signal sequences. And obviously, they need to be specific for each specific compartment. So signal sequences that target proteins to the endoplasmic reticulum for eventual secretion have one kind of design. Signal sequences or targeting sequences that target proteins to chloroplasts have a different, look different. So do mitochondrial targeting sequences and peroxisomal targeting sequences. So they're all distinct, but they have the thing in common that they are all fairly short, and they're almost always attached to the terminus of the protein, mostly the N-terminus, but in some cases also to the C-terminus. So one obvious problem in bioinformatics would be to take the amino acid sequence of a protein and look for one or other of these targeting signals to be able to predict just from the protein sequence where the protein is likely to end up in the cell. And this is called signal peptide or targeting protein sorting prediction. So I will specifically discuss one of these signals, namely the signal sequences that target proteins to the endoplasmic reticulum for transport out of the cell, for secretion out of the cell. And I will tell you a little bit about the history of the prediction methods, how they've been developed, and where we stand today in this field. So very early on, when the first few sequences of signal sequences of this ER targeting signal sequences became available, by just looking at them, it appeared from just a small number of examples that they seem to have a kind of a tripartite design with a positively charged N-terminal end, just a few residues long, with a hydrophobic central region, something like maybe 10 to 15 residues long, and ending with a more polar region leading up to the cleavage site, where an enzyme cuts off the signal peptide after it has served its purpose to target the protein to the endoplasmic reticulum. And looking more closely at this, around this cleavage site, it appeared that there was a small conserved sequence motif, namely that the residues right before the cleavage site and two positions upstream, namely in positions -1 and -3 relative to the cleavage site, these two positions seem to require a small side chain, an amino acid with a small side chain, such as glycine or alanine or serine, to direct the cleavage enzyme to the correct cleavage site. So this was kind of by eyeballing a few examples, early examples of signal sequences. One could kind of make a hypothesis that maybe this is some sort of conserved overall design of the signal sequences. One interesting aspect that was immediately clear from just looking at a few examples is that the precise amino acid sequence was not conserved. So you could have any collection of hydrophobic residues, hydrophobic amino acids in this so-called H region, and you could have either lysines or arginines in the N region, and they didn't have to be in a very specific position. So it's more this sort of overall design with positive charge, hydrophobic segment, and this cleavage region that was conserved, not the specific amino acid sequence. So that made it a little bit more difficult, obviously, to come up with good prediction methods, good bioinformatics-based prediction methods to find these N-terminal signal sequences. And I'll take you quickly through a couple of early methods just to show you how this developed. So one of the very first prediction methods was this one. And basically it was based on what we know from the previous lecture as a position-specific scoring matrix, a PSSM. So in this case, the way this was derived was that a collection of known signal peptides with known cleavage sites, all these sequences were aligned from their cleavage sites. So they had coincident cleavage sites between position -1 and +1. And then the amino acid counts in positions upstream of the cleavage site towards the N-terminal region were just counted. How many alanines, how many cysteines, how many aspartic acids, etc., in each position, right? And here you could immediately see, for instance, this -1, -3 pattern. So -1, you have lots of alanines and lots of glycines and lots of serines, but very few other residues. So that's clearly under selection, this position -1. And -3, similar thing, quite a lot of alanines, cysteines, a few isoleucines, quite a lot of serines and threonines, and valines that also have a fairly small side chain, but a lot of absent residues that were not found in this -3 position. And as you come into more towards the N-terminus, you see an accumulation of hydrophobic residues like leucine. You start to see them from sort of -4 perhaps, and then they really reach from -6 onwards, right? You get lots of leucines and lots of valines as well. These are also big hydrophobic residues, phenylalanines also. So you see this, and this is really too short to start to see the positively charged residues. So you don't see any here, no lysine and no arginine. But if you were to extend this a little bit more, you would start to see a few lysines and arginines appearing when you get towards the N-termini of these signal peptides. So this is just basic amino acid counts. These are the expected average frequencies of these amino acids in proteins in general. And so this was just as we did with the PSSMs. This was first converted to normalized by dividing the raw counts with the expected counts, and then taking the logarithm of that quotient to get what's essentially a PSSM for signal peptides. And now you can just take this matrix, you can scan it across the sequence, and for each position across the sequence, you just calculate the score from the matrix, and then you take the highest scoring position and you say, well, how high is the score? Is it high enough that we qualify as a signal peptide, or not. And you can also, from the highest scoring position, make a prediction for where the cleavage site is in the sequence. So doing that on a collection of known signal peptides, those are in dark, and here is the score, maximum score for each sequence, and a collection of proteins, N-terminal ends of proteins which were known not to have signal peptides in them. And you see that this simple PSSM made a pretty good job of discriminating between true signal peptides, all the black ones, and proteins without signal peptides near their N-termini. And you could sort of make a, you could say anything above this score, you predict a signal peptide, and anything below this score, you predict as none. And you see the overlap is quite small. So it was a fairly efficient method to detect signal peptides in protein sequences, and also to predict their cleavage sites. Now, this was the story here, the history here has developed pretty much along the lines that I talked about in the previous lecture, namely going from PSSMs to more complex models. And machine learning was applied very early on in this particular field as well. And the first machine learning method that was applied to signal peptide prediction was actually not hidden Markov models, but neural networks, artificial neural networks, ANNs. I'm not going to discuss ANNs here, and I think you heard about them in other lectures. So let me just say that an ANN is composed of an input layer, which essentially reads off the sequence of the protein, which you scan along and allow it to read off the sequence of the protein. Then you have hidden layer, or more than one. You can have multiple hidden layers connected to this input nodes, and then there's some processing going on in these hidden layer nodes, and eventually the signal makes it to the output layer, which is simply in a discriminatory task like this one. You have simply have an output layer that says, in our case, signal peptide or not signal peptide. So it's a discriminator, basically. So this basic neural network technology was applied to signal peptides. And of course, like in every machine learning or even any bioinformatics prediction context, you need a high quality data set, training data set, both positive and negative examples. That is to say, sequences that are not signal peptides and sequences that are known to be signal peptides. You have to decide on a ANN architecture. That is to say, you have to decide on the number of nodes in the input layer, on whether to have one or more hidden layers, and some fine-tuning of the architecture. And then all the internal parameters, just like in a hidden Markov model, all the internal parameters that describe the ANN are systematically optimized during a training session, where you use your training data set to essentially optimize these parameters. And you can evaluate the predictive performance using cross-validation normally. So this was done also in this example. And this has actually been a recurring project since many, many years. As you can see, this is an output from version 4 of the predictor called Signal P, based on neural networks. So it's been updated a number of times with bigger training sets and more refined neural network architectures. So here is just to show you the type of output you get from Signal P 4.0. This is the graphic output you get. Basically, the green shows you the predicted propensity for this part of the protein to be a signal peptide or not. So if this is higher than the cutoff here, you predict it to be a signal peptide in this N-terminal region. And the red and the blue, especially the blue, predicts the cleavage site. So you can see the summary down here. Because this is high, the prediction is yes, this is a signal peptide, and the predicted cleavage site is between this threonine and this glutamic acid, right in here. And looking by eye on this, you can see, yes, there is kind of a long N region here with at least one positive charge. Then you have a hydrophobic region. And then you have this cleavage region that goes from about here, from the threonine, up until the end of the, in between the threonine and glutamic acid. That's the predicted cleavage site. So you would also say by eye that this looks like a good signal sequence. So this is the type of output you get from a predictor such as signal P. Now the latest version of signal P is signal P 5.0. That's from 2019. And now you can see that the architecture in particular, the number of signal peptides in the training set also went up a bit, but mainly it was the architecture of the underlying neural network architecture that has been much more complex now. And it's using what's called deep recurrent neural networks. So this is essentially borrowed from deep learning methodology, you know, that Google and others are using now for predicting protein structure and for predicting this and that, for figuring out what you're like as a person and what your interests are and so on. So deep learning is basically neural network technology, but taken to a new level with much more complex types of neural networks. So this was tried also now in signal P 5.0 on the signal peptide prediction problem. Here is just the server. So you just paste in your sequence and hit the button and you get the prediction come back. But what's more interesting is perhaps how well this type of predictor performs now. So here is signal P5 compared to the performance of signal P4 and another one called DeepSig. So in blue, and also this is because there are different versions trained on bacterial signal peptide and eukaryotic signal peptides. So you have to, so you can see the performances differ a little bit between bacterial sequences and eukaryotic sequences. But by and large, you can see that this measure here, the blue measure, that's essentially a measure of how good the method is in identifying signal peptides without making a lot of over predictions, false positive predictions. And you can see this can reach 1 if the prediction is perfect. You find all the known ones and you don't find any false positive, you don't make any false positive predictions. And you see it's up to about 0.9 now. It's slightly better than signal P 4.0 and a bit better than this DeepSig. And you see it's almost as good for eukaryotic signal peptides. In orange, you have what's called cleavage site recall, and that is the fraction of cleavage site predictions that are correct. That is to say, if you predict a signal sequence and you also predict a cleavage site, how often is that cleavage site correct? Sometimes it can be mispredicted by 1 or 2 residues, but how often is it correct? And you see that it's correct in about 80% of the time for bacterial signal peptides and maybe 75% of the time for eukaryotic signal peptides. So they're not only good at recognizing signal peptides, but they're also good at predicting the exact cleavage site between the signal peptide and the mature protein. And finally, you have what's called a recall. And these are the fraction of real signal peptides predicted as correctly predicted as a signal peptide and with the correct cleavage site. So, and this is also especially for signal P5 and on bacterial proteins, this is now much better than the earlier ones. So this is, if you have 100 real signal peptides, what this measure is how many of them are predicted as signal peptides, and in addition, have the correct cleavage site. So these predictors are pretty good now, you can say, and they have for a long time, this one and earlier versions of signal P have been applied in genome sequencing pipelines, for instance, when you first predict all the proteins, and then you can process them through different bioinformatics predictors, such as signal P. to identify secretory proteins and also to predict where the cleavage site for the signal peptide is located. So this is done on genome-wide scales now as part of the pipelines for annotating newly sequenced genomes. So this was where the field stands kind of when it comes to predicting signal peptides that target proteins to the endoplasmic reticulum. I told you that there are other sorting or trafficking pathways in the cell. There are mitochondrial proteins that have their own specific type of signal sequence, chloroplast proteins as well. And in bacteria, there are also other types of protein secretion pathways that are based on different types of signal peptides. And there is now predictors have been developed based on the same kind of technology for identifying mitochondrial signal sequences, for chloroplast signal sequences, for these bacterial ones as well. And for the eukaryotic cell, these have even been merged into one protein targeting predictor called target P, which takes a sequence as input and tries all these different mitochondrial, chloroplast, and ER targeting signal sequence neural networks, and then weighs them against each other, weighs the output against each other, and comes up with a final prediction as to whether the sequence encodes one or other of these signal sequences or none of them. So this is kind of a combined or integrated predictor for localizing proteins in the cell. Okay, so that is the protein targeting problem, and where bioinformatics, what kind of contributions bioinformatics has made in this particular field. So let me now turn to membrane proteins. So membrane proteins are proteins that are designed by nature to live their lives not in the aqueous cytosol or in an aqueous compartment, but actually to be integrated into lipid bilayers as integral membrane proteins or transmembrane proteins. And here is an example in blue. This is a membrane protein unknown structure that has been simulated by molecular dynamics as when it's inserted into a lipid bilayer with water molecules on both sides. And so you see that the protein is well buried inside the lipid bilayer. It's almost completely immersed in the lipid bilayer, and it only makes small contacts with the aqueous environment on both sides. This happens to be a water transport, a water channel, and you can see that it actually does have a channel through which water molecules can pass through the lipid bilayer. But other membrane proteins obviously don't allow water molecules to pass. You can also notice in this MD simulation that water molecules almost never make it into the hydrophobic core of the membrane between the hydrocarbon tails of the lipids. There's almost no water molecules. So in order to sit or to essentially be energetically happy in an environment such as this, the surface of this membrane protein has to be very hydrophobic. It has to be composed mostly of hydrophobic amino acids that kind of dissolve themselves between the hydrocarbon lipid tails. So membrane proteins the membrane embedded parts of membrane proteins tend to be very hydrophobic, very rich, very enriched in hydrophobic amino acids. And it's only the parts that stick out on either side that have more polar amino acids and that can interact favorably with water molecules or with the polar lipid head groups, these yellow balls here. So that's what membrane proteins look like as when they're embedded in a lipid bilayer. Now, today we know the structure of quite many integral membrane proteins, and what we have learned from all these structures is that nature has really designed only two basic architectures for integral membrane proteins. And one is what's called the helix bundle architecture. That is the segments in the protein that span the lipid bilayer coil up into alpha helices, and then they connect with short loops to the next alpha helix and the short loop to the next one, and so on. So they go back and forth, weave back and forth across the membrane. And obviously these helices tend to be very hydrophobic, have a lot of hydrophobic amino acids in them, in order for them to be energetically favorable when located inside the lipid bilayer. But obviously, in the loops and the parts exposed to the aqueous phase, they can have polar and charged amino acids. So that's the helix bundle architecture: transmembrane, hydrophobic transmembrane alpha helices that pack together to form well-defined helix bundles. And they can create all kinds of transport pathways and ion channels and all kinds of things. The other architecture is called the beta barrel architecture, and it's based on beta strands that connect into a large antiparallel beta sheet that coils up, so that the most N-terminal strand in the beta sheet, which would be this blue one, and then you see how the strands go round, 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 and eventually the last strand in the beta sheet, this orange one, comes out here, and the first and the last strand pair up to essentially roll the beta sheet into a barrel. And here, every second amino acid, the ones that protrude towards the lipid bilayer, have to be hydrophobic in order to make this surface hydrophobic, so it can sit nicely in the lipid environment. But these are really the only two integral membrane architectures that nature seems to have come up with so far. So if we look a bit more closely, so here is the 3D structure of the kind I just showed you for a helix bundle membrane protein. So let's focus on those for a while. Now, if you kind of unravel the 3D structure here, you get what's known as the topology of the protein. So the topology of the protein specifies which parts of the protein chain are actually form these transmembrane alpha helices, which parts are embedded in the lipid bilayer, and which parts face either the cytoplasm or the extracellular space when the protein is integrated in the membrane. So it's kind of a secondary structure prediction problem, is to predict where are the transmembrane alpha helices in the sequence, and moreover, which way is the protein oriented in the membrane. That is to say, which loops and tails are oriented towards the cytoplasmic side of the membrane, and which are on the extracellular side of the membrane. So clearly, with this, if you know the transmembrane helices, there are two possible opposite orientations in the membrane, right? And you would like to be able to just take a sequence, predict where the transmembrane helices are using some bioinformatics method, and also predict the overall orientation of the protein in the membrane, so you know which parts are outside and which parts are inside. That would be a good prediction to have, even if you don't have the 3D structure, or especially when you don't have the 3D structure of your protein. So topology prediction has become, or has for a long time been, kind of a favorite area in bioinformatics. So to provide some background for how to best design a topology prediction algorithm, let's just collect some statistics on membrane proteins. So this is statistics on the typical lengths of the transmembrane alpha helices, taken from a number of known X-ray 3D structures. And what you can see is that most helices tend to be between, say, 20 and possibly 30 residues long. That's the majority of the helices of this length. And that makes perfect sense, because a membrane is roughly the hydrophobic core of the membrane, corresponding to the hydrocarbon tails, is about 30 angstrom across. So to span a width of 30 angstroms, you need a helix of the same length, obviously, and that's about 20 residues. So each residue adds another 1.5 angstroms to the length of the helix. So 20 residues long helix is about 30 angstroms long. So it makes perfect sense that it should be roughly from 20 and maybe up to 30 or so if it's if it gets longer, maybe it will start tilting in the membrane a little bit, and you don't want it to tilt too much. So there is no point in making it much longer than 30, let's say. So this makes perfect sense from what we know of the dimensions of a lipid binder and what we know of the dimensions of alpha helices. So typical lengths, say 20 amino acids, 20 to 30 amino acids. If we look more in detail, we find that there are specific amino acids, namely tryptophans and tyrosine, that are enriched in the regions of both the beta-barrel proteins and the helical proteins in the part of these proteins that sit in the lipid head group region. So this is at the transition point between the aqueous phase and the hydrophobic core of the membrane. And apparently both tryptophan and tyrosine have a special affinity for this transition region. And perhaps not surprisingly, because both tyrosine and tryptophan are aromatic, so they have a hydrophobic character, but they also have a polar moiety in the side chain. Hydroxyl group in the case of tyrosine, and an H group in the case of tryptophan, or sorry, a nitrogen, polar nitrogen atom in the case of tryptophan. So for that reason, they seem to be perfectly suited to sit in this interface where they can both interact on one side make hydrophobic interactions with the hydrocarbon tails, and on the other side, they can make polar interactions with lipid head groups and water molecules. So anyway, what it means is that transmembrane helices and these transmembrane beta strands tend to have tyrosines or tryptophans near their ends. So that's another sort of statistical regularity that might be useful for prediction purposes. If we measure loop lengths, how long are the loops between transmembrane helices? The answer is they tend to be short. So the full line here is the length distribution collected from a number of loops, and you see that it peaks around 10 or 12 residues, 10 to 15 residues length. So the loops tend to be short. There are some that are very long, and those very long loops tend to form independently folded units in either the cytoplasm or the extracellular side of the membrane. But those are fairly rare, and so most loops are really very short, just up to, say, 15 residues. And then the next helix comes along. And finally, there is a very strong bias in the distribution of positively charged residues, lysines and arginines, between the loops that face the inside of the cell and the loops that face the extracellular space that are on the outside of the cell. So the simple observation is, and this was first just a statistical observation, is that positively charged residues are highly abundant in the loops that face the inside, but are comparatively rare in the loops that face the outside, the extracellular space. So in bacterial inner membrane proteins, for instance, it's up to a 4-fold difference in the frequency of lysines and arginines in the loops facing inside compared to those facing outside. And you have a similar tendency in eukaryotic plasma membrane proteins, or even in these organellar membranes like the thylakoid membrane in chloroplasts or the mitochondrial inner membrane, you find a similar bias with positively charged residues being enriched on the part that's facing the inside compared to the part that's facing the outside. This is called the positive inside rule, and it turns out to be actually very useful for prediction purposes. Just to note in passing that it's not just a correlation between orientation and incidence of positively charged residues, but you can actually control the orientation of a membrane proteins by relocating positively charged residues from one side of the structure to another side of the structure. So here is actually an experiment. This summarizes an old experiment where a simple 2-spanning membrane protein, 2 transmembrane helices in the E. coli inner membrane, was re-engineered from its normal topology with both the N-terminus and the C-terminus facing the periplasm, facing the outside of the membrane, to one where it was turned on its head, basically, such that both the N and C-terminal now faces the inside. And this engineering, this reorientation of the protein was achieved by first removing positively charged residues from this highly charged cytoplasmic loop, leaving only 2. But notice there's none on the N-terminus, so it still has more in this loop than on the N-terminus. And even this deletion mutant oriented like the wild type. But then in the second step, 4 extra positive charges were added to this N-terminal tail, 4 extra lysines, inverting the charge polarity between the N-terminus and this loop. And when this version was expressed, it actually turned out to insert backwards with the loop now on the outside and the N-terminus on the inside. So just adding these extra 4 positive charges to the N-terminus was enough to redesign the protein to take the opposite orientation in the membrane. So clearly positively charged residues are not just correlate with orientation, but actually control orientation. Okay, so that was just an aside. Now, topology prediction. So what we want to do, we want to take a sequence of a protein, we want to feed it into some kind of predictor, and we want an output that tells us, okay, so here are the approximate locations of the transmembrane segments in this protein, the transmembrane helices in this protein, and these loops are on the outside and these loops are on the inside. And for now, this is restricted to helical membrane proteins. So I'll come back to beta-barrel proteins later on. Okay, so this is the problem. How do we construct a predictor that can do this trick? Take the sequence as input and produce a topology prediction as output. So let me just quickly rehash the 3 most important characteristics that we learned from just looking at doing some statistics of known membrane protein structures, that the parts of the helices that span the membrane are rich in hydrophobic residues, and they tend to be somewhere between 20 and 30 residues long. Very often, these helices end with a tyrosine or a tryptophan. Here you see a tyrosine, here you see a tryptophan. And the loops facing the cytoplasmic side, the inside, are rich in lysines and arginines, positively charged residues. And this you can see in this example, which is, in fact, I think the same water channel protein that we looked at in the molecular dynamic simulation. You see that really the tryptophans and tyrosine are definitely concentrated in this head group region, lipid head group region. The transmembrane helices are hydrophobic, and essentially all the lysines and arginines are on the inside, except one, which is here, that is actually part of the water channel through the membrane. So it's required for functional reasons here, but there is none in these periplasmic loops for this in this particular case. So those are the 3 characteristics that we think are universal, essentially, for all helical membrane proteins, and that form the basis for the topology predictions. So as I said, this has been a favorite field in bioinformatics for many, many years, and there is consequently a lot of different predictors around. I will specifically talk about this one in a minute or two, TM-HMM. These are the more modern ones. You can see they're based on HMMs, multiple sequence alignments and HMMs, or dynamic programming. And I'll also mention this one, which is a consensus method that basically takes predictions made by other predictors as input and tries to make a consensus prediction. Then there were an intermediate aged group that was not based on machine learning, and I'll specifically discuss TopRed, which was just looking for hydrophobicity and using the positive inside rule in a way I'll tell you about. And then there were some very early ones that were only based on hydrophobicity, because they were designed before this positive inside rule was known. But these were very poor. They were very bad predictors. These were reasonably good predictors, and these are even better predictors. So I'll show you TopRed because it explains very nicely how the positive inside rule can help in topology prediction. And then I'll focus on TM-HMM, and I'll just mention TopCons briefly. So TopRed was really the first method that took both hydrophobicity and the positive inside rule into account. And the way it worked was designed was the following. So what you see here is a hydrophobicity plot for a inner membrane protein from E. coli called lactose permease, or LAC-Y. So a hydrophobicity plot is simply you take a window, say 21 residues, typically the length of a transmembrane helix, and you place that across the first 21 residues in the protein, and you calculate the average hydrophobicity of those 21 residues using some hydrophobicity scale. And there are hundreds of hydrophobicity scales, but they all correlate quite a lot to each other, so you can use any one of them. So that gives you the average hydrophobicity of the first 21 residues. And you plot that as the first point on this curve. Now you shift by 1, so you go from residue 2 to 22, and you repeat the calculation, calculate the average hydrophobicity, plot the value, and then you continue scan along the sequence until you reach the C-terminal end. So obviously, if you hit a segment that's roughly 20 residues long and has lots of hydrophobic residues in it, it's going to have a very high hydrophobicity. So it's going to define a peak here. And if you hit, say, a loop region in between transmembrane helices, it's going to have much fewer hydrophobic residues and instead have polar and charge residues. So you have a low average hydrophobicity. So you would expect for multi-spanning inner membrane protein, you would expect to see a number of peaks corresponding roughly to the transmembrane helices, right? Now, what you want to do here is you want to predict which of these peaks actually are true transmembrane helices. And so you would like to be able to define a cutoff value, say this line here, such that would clearly discriminate between peaks that correspond to transmembrane helices and peaks that are not quite hydrophobic enough to become transmembrane helices. If you could find such a cutoff, then this problem would be solved. You could immediately tell, okay, these are the transmembrane helices, this is how many they are, and the only thing left remaining would be to predict the orientation of the protein. But it turns out that's not possible. So if you try to do this, what you quickly realize is that you cannot find one cutoff that will cleanly distinguish between true transmembrane helices and peaks that are not hydrophobic enough to correspond to transmembrane helices. What you can do is you can find a cutoff that you push high enough that you can be pretty confident that anything above this cutoff is indeed a transmembrane helix. So that's the upper line here. But the price you pay is that there will be some peaks like this one and this one and this one, which will fall below that cutoff, but still might be considered potential transmembrane helices. And you wouldn't be able to tell whether or not it is. So what you can do now, or what was done in TopRed at this point, was to define a second cutoff value that was pushed downwards so far that any peak falling below that cutoff in a collection of known membrane protein topologies, that any peak falling below it was highly unlikely to be transmembrane, 99% likely not to be a transmembrane helix, right? So that's fine. So then anything below this lower cutoff you can ignore. It's not going to be a transmembrane helix. Anything above the upper cutoff you can be pretty confident will be a transmembrane helix. But then ever so often in some protein like this one, there are peaks that are in between the two cutoffs, and you can't really tell then whether or not this is a transmembrane helix. So this is where hydrophobicity analysis by itself left you, basically. Now, if you know about the positive inside rule, you can actually improve things quite a bit, because what you can do then, or what TopRed did, was to say, okay, so I take all the peaks that are above the upper cutoff. I say these are for sure transmembrane helices, and there are 11 peaks here. So I would say, okay, there's at least 11 transmembrane helices in this protein. But then I have this intermediate peak, and it leaves me with two possibilities. Either it is not a transmembrane helix, in which case I only take the 11 and predict this as a loop, or it is a transmembrane helix, in which case I predict this as transmembrane and get 12 transmembrane helices, right? And those two predictions are here. So here there is 11 helices, and this peak here is predicted to be part of a loop. So it's 11 transmembrane helices, and here is 12 transmembrane helices. So now this peak is predicted to be a transmembrane helix, right? And now the problem is reduced to choosing between these two models, which would I put my money on? And the way I choose, or the way Topred chose, is to basically look at the distribution of positively charged residues in the loops in the two models. So here, this is the number of lysines and arginines in the loops on either side. And so start with this one. It's 2 here, 1, that's less, 3, 0, 5, 0, 4, 1, 2, 3. So up to this point, it all looks fine, right? Many positive charges on this side, few on that side. So you would say, yeah, this looks like a good topology. And moreover, you would predict this to be the inside, facing the inside, facing the cytoplasm. But then you get to the end here, and now suddenly you have 3 here and none in this final loop, and 2 in this tail. So this doesn't look great, right? And if you just sum up the difference in the number of charges, this side minus that side, you find the difference is 9 positive charges. The net bias is 9. Now you take the 12 TM model and you count the charges, and now what you see is that it looks fine from beginning to end. There's more charges on the inside than on the outside throughout the whole protein. And the charge bias, the difference between these positive charges and the sum here, is actually 17. So knowing about the positive inside rule, you would, in this case, you would definitely put your money on this alternative and not this one. And of course, for lac permease, this is the correct one. It has 12 transmembrane helices, and it has both N and C termini facing the inside. So what was difficult to do when you only looked at hydrophobicity actually turned out to be quite simple when you could also take the positive inside rule into account. So that's what Topred did. Run hydrophobicity plots, identify all the high peaks that are certain to be transmembrane helices, identify all the and identify any peak in between the two cutoffs, and then construct the possible topologies. I mean, if there is no peak here, there is only one. If there are 2 peaks, then of course you have more possible topologies. And then in the end, you rank them based on this charge bias, and you pick the one with the highest charge bias. So that really improved prediction, topology prediction, by a big leap, going from just what you could do just with hydrophobicity. But of course, it's a very, very simple model, and there's every reason to expect that one could do better. And indeed, one can do quite a bit better. So this is where, again, machine learning comes into the picture. And in particular, I'll talk about this method called TMHMM. So it's based on a hidden marker model approach. So if you remember from when we talked about hidden marker models in the previous lecture, what you have to do to set up, build a predictor based on hidden marker model is you have to first design the architecture of the HMM that you then want to train on some kind of training data set. So here is the architecture that underlies the TMHMM predictor. So the idea is that if you just look at a linear sequence of a helical transmembrane protein, you have the N-terminus itself. It can be either inside or outside. Then it's necessarily followed by a transmembrane helix. So if you were to say start on the outside, the helix will take you to the inside. So by necessity, there would be a loop on the inside following the helix. Then there is another helix, and then by necessity, the next loop will be on the outside, and so on. So this can be roughly captured in a hidden marker model architecture, such as this one, where you have a central helix state. So these would be states corresponding to residues that are in the transmembrane helix itself, buried in the core of the membrane. Now, helices have, as I showed you, they have tyrosines and tryptophans on their ends. So you might want to capture that by having a special cap state that on either side of the helix. So the helix would have a lot of hydrophobia. The emission probabilities from the helix would be mainly for hydrophobic residues. The caps would have high emission probabilities for cap states for for polar residues, especially for tyrosines and tryptophans. And then you would have loop states. You would have a loop state on the inside, and obviously that would follow upon a helix and a cap. And obviously this loop state would have high emission probabilities for positively charged residues. When loops become very long, this positive inside, this high fraction of positively charged residues tends to get diluted. So if you have like a big globular domain between two transmembrane helices that are located inside or outside the cell for that matter, they will have a more balanced amino acid composition. So they will have a different set of emission probabilities. And then for technical reasons, it also turned out to be a good thing to distinguish between short loops and long loops and globular states on the outside of the cell. So this was the final hidden Markov model. You can go from, say, an inside loop. There is a transition probability to go to a globular domain and then back to the loop. And then there is a certain probability to go to a cap state. And from the cap states, you can go to the helix states. You have to go to the helix states, basically. From the helix states, you have to go to the cap state. And then you have some options here on the outside, short loops and back to the cap, long loop and back to the cap, or short loop and globular, or long loop and globular, and back again. And then back again to the cap. And then you have to go to the helix, and then you have to go to the cap, etc., etc. So this was the basic architecture. The core states and these loop states had to be modeled in more detail. So the helix core states, this is the internal structure for the helix core state. So you have a number of states that each can emit a residue, mostly hydrophobic residue then. But you also have delete states, so you can model anything. You can model any helix core that is from, say, 1, 2, 3, up to 25 residues long. So that's the longest possible core you can model. Or you can go 1, 2, 3, and then you can basically make a very long, you can make a huge deletion here and end up 24, 25. So 1, 2, 3, 4, 5. So in principle, you could even model a helix core that's as short as 5 residues and anything in between. So of course, all these transition probabilities have to be defined during the training, or estimated during the training, as you have to estimate these transition probabilities along the main path here. So that's the internal design of the helix core. The emission probabilities are all the same from all of these states, so they're tied, as the term says. So there's just one common set of emission probabilities from these states. Now here's a cap state. The cap state is 5 residues long, and you have to go through them one by one. But then you get to the loop state. And so the loops can also be of different lengths. And so you can see you can go from a cap, just a 1 residue in the loop, and then go back to the next cap and into the helix. Or you can go from the cap into a loop that can be as much as 20 residues long, right? But all of these states are also tied, so they have the same emission probabilities. But of course, you add a lot of parameters here, a lot of transition probabilities to model these varying loop lengths. And then you have the globular domain, which is just one state that can loop back to itself. So you can model globular domains of any size, essentially. And there is just one set of emission probabilities from the globular state. So this is how the underlying architecture of TMHM was built. And then a large number of transmembrane proteins with known topologies were used to train it, basically to estimate all the parameters. And finally give you a prediction method. So here is what the typical output looks like from TMHM. So what's done here is you take a sequence, in this case of this protein here, you feed it into TMHM, and it basically tells you what's the most likely path through the HMM taken by this sequence, or to generate the sequence. What's the most likely path of states to generate the sequence of GLP-T? And that is essentially shown here on the output. So the prediction is that the protein starts on the inside, which is this blue, signifies inside loop. Then there is a transmembrane helix, then it's an outside loop, another helix, an inside loop, etc., etc. And here you can actually see the probabilities for this loop to be inside is 0.9. Probability that this is a transmembrane helix is actually 1, and then that this is on the outside is also around 0.9, etc., etc. So you can, not only do you get a predicted topology, but you also have, by looking at these probabilities, you get a feel for how reliable the prediction is. So TM-HMM and similar methods, and there are quite many now, are turn out to be quite good to predict membrane protein topology. I also said that there are, as in many areas of bioinformatics, consensus predictors tend to improve things a little bit. So basically the idea is that you submit your sequence to a number of different servers that each run their own, in this case, topology prediction method. And then you collect all the outputs, and then you have a neural network, usually a neural network that is trained to basically weigh together the different outputs in the best possible way and come up with a consensus type of prediction. And that is what TopCons does. You see it was made by Arne Elofsson's group. And TopCons is a very useful tool because it gives you a consensus prediction, but it also gives you a quick display of all the individual predictions coming from the different servers. And so, for instance, here you can see that for this particular protein, I don't know which one it is, all these 5 servers give the same prediction, right? So that makes you kind of trust the prediction more than if certain, you know, if the results would be all over the place here, and one would predict 8 and another would predict 5 and another would predict 6 TMs. But even in that case, this helps you because maybe you will see then that it's in this area that they disagree, whereas this area they all agree. So then you can feel more confident of the prediction in this area than in that area. So how good are these topology predictors for helical membrane proteins? They're pretty good, actually. If you only ask them to discriminate between soluble proteins and membrane proteins, don't ask for the topology, but just, is this a membrane protein or not? Both the sensitivity and specificity are very high. So they're very useful in, say, taking a genome sequence, all the predicted protein sequences, and basically predicting which are integral membrane proteins and which are not. These predictors are pretty good at that. Correct topology, well, if you use single sequence predictors, that is, you just feed one sequence into the predictor, they give you the correct topology, say 70-75%, for 70 to 75% of the proteins. It's a little better for bacterial proteins, a little worse for eukaryotic proteins, but in this range. Some of them actually do a multiple sequence alignment before they do the actual topology prediction, and then use the multiple sequence alignment in some form as input to the predictor, and that boosts performance by maybe 10% or so. So if you can base your prediction not on a single sequence but on an MSA, then you can get better topology models. So these are good enough to be quite useful to many people, actually. So let me just say a few words about beta-barrel membrane proteins. It's not nearly as much work has been done for various reasons, but there are some methods to predict these beta-barrel structures as well. And of course, there is HMM methods, and this is just one example to show you the design of, or the architecture of the underlying HMM for this one here called GMBB2. So there is a model for the beta strand, the transmembrane beta strands. And remember I said every second residue needs to be hydrophobic. So it actually distinguishes 2 different states. It has 2 different states within the membrane span. These are the membrane exposed and the ones that are projecting inside the barrel instead, that are not exposed to the lipid bilayer. So they have different emission probabilities. So the white ones have one set of emission probabilities, and the gray ones have a different set of emission probabilities. And then in order to model different lengths of beta strands, you have the same possibility to make deletions, basically, delete states here. And then you have loop states, pretty much as in TMHMM, with an extracellular loops that can be anything from just one residue up to whatever this is, 12 residues. The same on the inside, and then you have globular states that loop back to themselves here. And so same procedure, you collect as many examples as you can, you train on them to get all the parameters, and then that's your predictor. These are not quite so good as the in terms of performance as the helix bundle predictors, though. So finally, let me say a few words about 3D structure prediction, because obviously, ultimately, this is what you want to do. You want to be able to take your sequence of your membrane protein and actually predict the 3D structure, not just the topology. And there has been, as you may know and have heard in this class, there has been quite impressive progress in 3D structure prediction in later years, which is based mainly on the fact that there are so many sequences now available in the database. So almost any protein you care to look, you're interested in, you can find hundreds, if not thousands, of homologous sequences in the same family, and this has been the key to efficient 3D structure prediction methods. And the basic idea that seems to have worked here is to exploit what's called co-evolving residues or correlated mutations in these big multiple sequence alignments. So imagine that you have, say you have 500 homologs, you do a multiple sequence alignment, and then you start looking for correlations between columns in the multiple sequence alignment. And you look for correlations of the type where, say, you see that this residue, if this is mostly, say, alanine in most cases, but you also find sequences where this is a serine, and then you look in all the other columns and you discover that whenever this is a serine, this residue over here is a glycine, whereas whenever it's an alanine here, it's a serine here. So those are correlated mutations. Apparently, if you mutate from alanine to serine, you have to make a compensatory mutation here from serine to glycine. And this indicates, doesn't prove, but it indicates that these two residues are actually close in space in the folded structure, such that if you mutate one, you have to make a compensatory mutation in the other one, or the 3D structure will not be well packed anymore. So that's the basic idea. And then there is a lot of advanced statistics behind here to filter out the ones that really are, can be explained by close contact, and those that are kind of just indirectly caused by close contact. But this is a problem that has largely been solved. And so what you can do when you have a big enough alignment is you can identify a number of these coupled sites, which basically means you can say, okay, so I know now that this residue is close to this residue in the 3D structure, this one is close to that one, and these two are close, and these two are close, and these two are close. And turns out you don't need very many of these constraints, of these close approaches, in order to constrain the number of possible 3D folds to essentially just one or a small number. So with these evolutionary couplings, as they're called, you can actually come up with very good 3D structure predictions. This is quite similar to how you solve structures with NMR experimentally, because there you can measure distances between two nuclei. And if you have a number of these pairwise distances, you can then basically fold your protein while observing these distance constraints. So it's essentially using the same methodology as that. But you can also use a lot of more advanced sort of machine learning superimposed on this that takes all of this sequence variation that you have from the multiple sequence alignments into account. And these methods now are actually surprisingly good. And you can, in many cases, you can get very close to the actual structure. using these types of 3D structure prediction algorithms. The only caveat is you need a fairly large number of homologs for this to work. But with the enormous rate of sequencing happening today, this is a problem that will soon be solved for almost all protein families, membrane protein families, or families of soluble proteins for that matter. And here are just some examples of experimentally known structures. I forget which is gray and which is blue, but one is the predicted one using one of these methods, and the other one is a known X-ray structure. And you can see that in these cases, they are many of them are extremely close to the real structure. And so you can really start to trust these predictions quite far now. So with that, I'd just like to summarize. I talked about signal P and target P, signal peptide, targeting peptide predictions. Noted that there are two architectures for membrane proteins, helix bundle and beta barrel. Talked about the positive inside rule, about topology predictions like TopRed, the old original method, and the HMM-based TM-HMM, and another HMM-based method to predict topologies for beta barrel proteins. And then finally, I mentioned this 3D structure prediction by co-evolving residues that you might meet in other lectures in this class as well. So with that, I'd like to thank you for staying with me for these two lectures, and I hope we can have a good discussion. Thank you very much.